Okay, what about animal models in the development of drugs and vaccines? Well, just three very quick examples. Anti-rabies vaccine developed in about 1885 by Pasteur using dogs and rabbits. The point about the use of the dog here is that rabies is a virus which affects both humans and animals, and therefore animals could be a model for developing a vaccine. And in fact, having developed a vaccine against rabies, which was effective in dogs, Pasteur found that the same vaccine was equally effective in humans. So we now have a whole lot of vaccines that have to be tested still in animals. And until such a time as we have alternatives, we will have to continue to use these animals to test the safety of these vaccines. For example, polio is a live vaccine and it's notorious for reverting to neurovirulence. If we did not test it in animals, then every so often we would give a batch of polio vaccine to children who would then get polio. And as a result of that, people, of course, would stop vaccinating, they would lose confidence in the vaccination process, and polio would come back as a serious disease.
In this episode called Biologicals, Vaccines and NIBS, we will be looking at two sources of information. The first is from the Canadian Council on Animal Care's website. You can see it in front of you, and if you look at the third column, you will see that there's 4 stages or 4 parts of how vaccines are tested. The first one, which is highlighted here, says vaccine potency, veterinary use. The second one says vaccine potency, human use. The third one says vaccine safety, veterinary use, and the last one says vaccine safety, human use. We will be looking at all 4 of those areas, as well as referring to the BBC Radio 4 programme called Secret Science, which looked at the National Institute of Biological Standards and Control, otherwise known as NIBS. I don't like repeating material, but in this case I feel I have to. This is the definition of biologicals from the last episode. I can't really assume that people have seen or heard that episode, so please bear with me as I repeat the definition of biologicals. I'll probably have to do it in the next episode as well.
We deal with biological medicines.
Stephen Ingalls, Director of the National Institute for Biological Standards and Control.
Biological medicines are medicines made from cells, from tissues, and they are different from normal medicines because they're much more complicated. They're much more complex in their structure. A biological medicine would be, for example, a vaccine that's made in cells. And that would be hugely more complicated than, say, paracetamol. And because of that, it needs to be treated differently, needs to be analysed differently, needs to be measured differently.
And measuring biological medicine is critical. You can't just count tablets or weigh a medicine. You have to be able to measure how active it is, and to do that, you need to compare it against something of known activity, a standard.
So here we are at the first slide, which reads vaccine potency, veterinary use. So the conventional test method is the lethal challenge test in the batch potency testing of inactivated swine. I think you pronounce this Erie C. Pellis vaccines. So that's the conventional test, and in the name and description of the alternative test is the stereological method for the batch potency testing of inactivated swine Erie C. Pellis vaccines. The description of the test reads, this test uses an alkali extract of E. aureosyopathiae as the coating antigen to detect protective serum antibodies of immunized mice. Serum antibody levels are estimated by comparing the titration curves of the test serum and reference serum. So looking at this test, the first problem I'd have with it is it looks like if this is a swine virus, a pig virus vaccine that they're testing on mice, which, you know, you're going to have interspecies differences there, so that could cause a problem. If it had been tested on pigs, there's no scientific argument against the test because you won't have interspecies differences, but this seems to be tested on mice, so there might be. Anyway, it says the test has validation status, was validated by ECVAM, which is the European Centre for the Validation of Alternative Methods, and that was in 2002. It says it has got some regulatory status. It's included in the Pharmacopoeia Europe, is that monograph? I'm not sure, but PHEUR monograph. So it does have some regulatory status, and the effect on animals, it says, is a reduction or a refinement because it does not rely on death as an endpoint. So obviously the conventional test method at the top of this slide, the lethal challenge test in the batch potency testing of inactivated swine Erie C. Pellis vaccines, obviously is a lethal test, probably is some kind of LD50 test.
For more than a century, the mass of a kilogram has been fixed by a single cylinder of metal sealed inside 3 locked safes at the International Bureau of Weights and Measures near Paris. It's used to calibrate the unit's mass precisely, and if you wanted to make sure that your kilogram of apples really was a kilogram, you could check back with this internationally agreed standard. But biological standards are different. No matter how many times a reference kilogram is accessed, it's undiminished. Not so for biological standards. In comparing them with what you have to actually use up your little standard sample during the testing process. This means the Institute has to prepare tens of thousands of ampules of each standard, which can then be made available to those who require them in the development or testing of biological products. Every batch of a particular medicine made anywhere in the world can be measured directly against NIBS standard.
So now we are going to look at vaccine potency human use. In the left-hand column of this slide you can see conventional test method, and the first one reads lethal stroke paralytic challenge test in the batch potency testing of tetanus toxoid vaccines for human use. The 4 tests in this slide read lethal stroke intradermal challenge test in the batch potency testing of diphtheria vaccine. The next one reads combination tetanus and diphtheria vaccines, separate serology tests for each vaccine. The next one reads potency test of hepatitis B vaccine, brackets mouse. And the last one reads serological potency test of poliomyelitis inactivated vaccine, brackets rat. And this last slide reads lethal challenge test for rabies vaccines. In the second column we can see the alternative tests. So the alternative test to the serological potency test of poliomyelitis vaccine is the antigen quantification. So we'll have a look at that because that will give a response to Dr. Michael Festing's introduction to this piece. And he also mentioned the rabies vaccine. So again, let's look at the alternative test for that. And again, if we look in the second column, we can see that there are 2 alternative tests for the lethal challenge test for rabies vaccine. The first one reads single dilution assay, and the second one reads humane endpoints for rabies potency testing. So let's have a look at all 3 tests now. So here we are looking at this slide. At the top it says conventional test method, as we said, serological potency test of poliomyelitis, and underneath that it says alternative test method, name and description, antigen quantification, as we have said. Validation status, it says no information, so that's not very good, but under regulatory status it says it has been accepted in PHEUR, which I think is Pharmacopoeia Europe or something. So it does have some regulatory status, and it says the effect or potential effect on animal use is replacement, which is very promising indeed. So unfortunately I can't really give you much more on this particular test, but it does give the impression that there is an alternative to using animals. Now we're looking at the alternative test to the rabies vaccine, the lethal challenge test for rabies vaccine. The alternative test method says single dilution assay. It says enables the analysis to determine that the vaccine has a potency significantly higher than the required minimum, but does not give full information on the validity of each individual potency determination. Under validation status, it says no information, and under regulatory status, it has been accepted in the PHEUR. Under effect or potential effect on animal use, it says reduction, so this obviously isn't a full replacement test. So now let's look at the second test, a second alternative test to the rabies test. And this one, that is the lethal challenge test for rabies vaccine. And under alternative test, it says humane endpoints for rabies potency testing, observation of clinical signs and application of an endpoint earlier than death to reduce animal suffering. Validation status, it's an initiative in the US. Regulatory status, it has some regulatory status in Europe and in the US. The effect or potential effect on animal use is refinement. We can see this as it is a classic refinement test, this one. Basically, they are looking for signs of weakness in the animal, heavy breathing, lethargy, maybe slight signs of convulsion, that kind of thing, rather than, you know, the ultimate death, the slow ultimate death of 50% of the test subjects. So regarding the polio vaccine testing for potency, it does seem like there is a full replacement. However, with the rabies vaccine, it seems like there's not a full replacement at the moment. However, in the future, surely this could be made possible with encouragement and money to the people at the National Institute of Biological Standards or other scientists elsewhere.
The scientists and expertise at NIBS may be unknown to the British public, but that would change in the event of a flu pandemic. NIBS is the front line as far as defence against the flu virus is concerned. The WHO go directly to NIBS with the highly dangerous strains of flu that might lead to a catastrophic pandemic. Less dramatically, NIBS also plays a key role in seasonal flu vaccines. Stephen Ingalls again.
We do an enormous range of different things here. One very important task is in influenza specifically, where we are responsible for producing the standards that the manufacturers require each year to make sure that their vaccines are appropriately potent. So in other words, the manufacturers need to measure how much is in each vaccine batch, and they need materials to help them do that. We do that. We do it for seasonal influenza every year. But then, on top of that, we help with pandemic preparedness. So it's our job, along with a very small number of other labs around the world, to obtain new strains of influenza that may cause a pandemic and to turn them in our labs here under high containment into safe viruses that can be distributed to manufacturers so that they can start to make vaccines.
I went to find out more about the flu and other virus work that goes on at NIBS. The virology division is headed up by Phil Miner with Jim Robertson and Una Dunleavy.
Seasonal flu is an extremely complicated business. It changes every year. There are changes in the vaccine every year simply because the virus drifts and different epidemic viruses appear. You have to make sure that the vaccine matches the epidemic virus that's going to arise, if it's going to arise, so you have to make an intelligent guess about that. You have to identify the right strains for the manufacturers to grow, and frankly it's a long and very, very complicated business, a little bit like turning an oil tanker.
So Una, when the strains have been selected, what's your role?
Once the decision's been made, manufacturers have got quite a short period of time to make vaccine, and in order for them to do that, we have to prepare some standard preparations so they can calibrate vaccine, so that they've got the right strains in there at the right concentration. We can do the testing within about 2 to 3 weeks so that we can get the reagents calibrated so that we can have vaccine in the shops and the pharmacies for the season in October.
So you have a double role effectively, because first of all you have to make all the standard preparations, then you send them out to the manufacturers, and then presumably they then send their things that they've made back to you to test them again to make sure that they're right against your standards.
That's correct, because whilst we have a responsibility for making the reagents, the calibration reagents, we then have vaccine presented back to us for testing for use in the UK. So we have a very short turnaround time for testing flu vaccines because of the need to actually make sure that supplies are available.
And we've made it sound quite easy, but actually growing flu is quite tricky, isn't it? Because some of them are rather reluctant. The strains that you might have in a particular year might not work well together, or they might not grow very well, and that some of them are quite difficult, aren't they?
Yes, some of them are difficult to grow. If a vaccine strain is found to be not very good at growing, then we have the technology here to actually improve those parameters. So we are involved in that as well.
We're now going to look at vaccine safety veterinary use. In this slide we can see the conventional test method is target animal safety test. So at least this will have some scientific validity to it, because it's actually looking at the target animal that the vaccine is going to be used for. In the name and description of the alternative test it says deletion of test. So let's have a look at the description of that. Yes, as I thought, deletion of test is what it actually says. Deletion of the test. It's no longer needed anymore. Under deletion of test it says a target animal safety test can now be waived when a sufficient number, e.g. 10, of batches have been found to comply with the test. I can't completely understand what this means, and I haven't got anyone that I can ask really, but it seems that this doesn't need to take place, this test, anymore, this target animal safety test, which is good for the test subjects that would have been involved. And anyway, it says under validation status, it says retrospective data analysis conducted by ECVAM. So maybe they have the information that they need to compare already. That's the only explanation I can find. Anyway, under regulatory status it says it is included in the PHEUR.
Seasonal flu is a year-on-year job for NIBS, but pandemic flu, the type that would spread like wildfire across the globe, is something else and calls for special powers. And Jim Robertson. Now, Jim, where do you come in? You're Mr. Flu here.
Or some people say Dr. Flu. My involvement is more at the level of research and development, and Una and my colleagues take very good care of all the seasonal vaccines and reagents that get prepared here for helping out industry. But as you probably know, every 10 or 50 years along comes the big influenza that sweeps around the world, and the one we call the pandemic virus. And these strains come in, we believe, from the avian world, where there are lots and lots of viruses out there. And you've all heard of bird flu, and this is being used as a term for describing the H5 avian flu that surfaced a few years ago in the Far East and is running rampant around half the world. And there's a huge concern that this is a possible pandemic influenza of humans. Now, normally we would deal with preparing a vaccine strain from this type of virus in the same way as we do for seasonal viruses, that we're dealing with it every year. But there's one difference here. This current bird flu virus, the H5 one, is really quite deadly, and everyone is quite aware of this. Its mortality in humans is currently running at over 50%. And that's a virus that is basically too hot to handle.
For the safety of the public and scientists alike, work on these too hot to handle viruses has to be undertaken in top security labs called Level 4 containment facilities. NIBS has several of these, in which it uses pioneering science to calm the most virulent tiger of a virus into a tame pussycat.
Over the last 5 or so years, new technology has been developed which we've imported into the division here, and we use that technology to calm down this highly deadly H5 virus. This is a technology that we call reverse genetics. It involves taking the virus apart, stripping it down to its bare genes, so to speak, cutting out that little bit of the gene that makes it highly virulent, stitching it all back together again, and recreating a virus which looks the same and in certain way acts the same, but is no longer deadly. In this way we can create a vaccine strain that can be handled relatively easily, and people don't have to take super precautions and dress in spacesuits and this sort of thing, because you couldn't make a vaccine with a virus that you had to handle under those circumstances. So we can tame the virus, and this is what we did quite effectively.
So you have a crucial role in taming the virus so that manufacturers can then prepare a vaccine. And in the past, that's actually taken quite a long time. But you've now got that time down to an extraordinarily small amount.
When we first began this work, generating a tame virus from a highly pathogenic avian strain took quite a number of months to achieve. It involved working on the virus itself to depathogenize it. It involved testing that virus, ensuring to the world and to the vaccine manufacturers who had received such a vaccine strain that the virus was in fact safe to handle. Over the years we've been able to refine this process, and this is what we've done. And we've brought that timeline down to under 3 weeks for generating the virus. And we hope that within a week of generating such a virus, we could actually send it out with an adequate level of assurance that this virus is safe to handle and safe to work with.
And those weeks might count in the face of a pandemic.
In the face of a pandemic, every week would count, every day would count. It's going to take time for vaccine manufacturers to make vaccine. There's no doubt about that. But once vaccine starts rolling off the press, every day, every week, they can make more and more and more vaccine. The sooner they start, the sooner they can get vaccine out onto the market and basically flood the market with vaccine.
So is this work on pandemic flu being replicated anywhere else in the world?
Due to the highly specialised nature of the procedures we go through, very, very few laboratories in the world are capable of doing this. And when we first started this in 2004, there were in fact only 3 laboratories, 2 in the United States and ourselves here at NIBS who had the capability. In the USA at the time, and for a long time, bureaucratic reasons prevented them using their viruses outside the USA. So it resulted in us and us alone having to supply the rest of the world, the rest of the vaccine manufacturers in the world with a virus that they could use to make bird flu vaccine. It still remains much that way. An awful lot of the focus is going to be on the work that we do here come a pandemic.
So you are uniquely important here in South Mimms to the fight against pandemic flu.
I think that's true to say, yes. There's very few of us can do this kind of work, and it is quite a unique situation.
We're now going to look at the last area of vaccine testing, which is vaccine safety, human use. The 3 tests on the Canadian Council for Animal Care, there are probably more tests, but these are the ones that are covered on their website, are abnormal toxicity test for human use vaccines, oral polio neurovirulence test, monkey intracerebral, and lastly residual toxicity in diphtheria vaccine (brackets guinea pigs). So they're the conventional tests, and now we're going to look at their equivalent alternative tests. The first description of the alternative test for the abnormal toxicity test for human use vaccines says deletion of tests. So it has said before, if it's deletion of tests, it's not used anymore. And under regulatory status it says accepted on condition of demonstrated consistency. So I can only imagine that they are comparing it with retrospective data, i.e. data that they already have, so there's no longer any need to do any more tests. And under effect or potential effect on animal use it says replacement, so this is a full replacement test, which is really excellent. Okay, let's have a look at the next one now. So the conventional test is the oral polio neurovirulence test, monkey intracerebral. The alternative test method is mutant analysis by PCR and restriction enzyme cleavage. PCR is used to measure the abundance of neurovirulent mutant sequences with a point mutation in the 5' non-translated RNA region of type 3 oral poliovirus vaccine. Increased ratio of 472-U to 472-C in type 3 OPV RNA is associated with greater success of the vaccine. Any type 3 vaccine batch which fails MAPREC should not be tested further in monkeys. However, if the vaccine is normal, i.e. negative, it must still be confirmed in a monkey neurovirulence test. So it seems that Dr. Michael Festing was probably right in his introduction. Even for, well, oral polio, it does seem like monkeys are still used to test the vaccine's safety, which is very depressing. Anyway, this is a test with effect or potential effect on animal use is a reduction. The validation status, the WHO, the World Health Organization, says this test is validated for type 3 oral poliovirus vaccines. So, yes, like I say, it's a bit depressing, and hopefully someone can come along and get a complete replacement test for the oral polio neurovirulence test. The last alternative test method we're going to look at is called the Vero cell test of diphtheria toxoid vaccines, and it's an alternative to the conventional test residual toxicity in diphtheria vaccine brackets guinea pig. It says Vero African green monkey kidney cells and MTT dye is added to wells containing either bacterial or cultural filtrates or reference diphtheria toxin. Vero cell viability is measured by observing the absorbance at 570 nm. I'm not sure what NM stands for, but we can see with this test it's not a complete non-animal test because it's using the African green monkey kidney cells. But it says the regulatory status is accepted into the PHEU, and the effect or potential effect on animal use is replacement. So that is absolutely great. So it does seem like there is progress being made, and if there was the will, the strong will there, I think all of these vaccine tests could be replaced with non-animal methods. Whether that will happen or not, I'm not sure.
The fear that the highly pathogenic H5 avian influenza might evolve so that it can spread from human to human is ever present. And whilst health fears are often hyped, this one is not. It's NIBS who will play a key role in deactivating the highly dangerous virus, making it safe enough for vaccines to be manufactured. If pandemic flu comes to pass, and a pandemic is well overdue, NIBS tiger taming and their ability to do it in such a short time will mean the difference between life and death for millions of people, not just in Britain, but worldwide.
We can hold our heads up with basic research in the research world. At the same time, we can turn that research to practical applications to helping with direct health topics. And for example, creating vaccine strains in the event of a pandemic. So a direct application of your research to helping promote the health of the world. And all that has come about because of the nature of the work of the Institute.
Jim Robertson there, the man who stands between us and a flu pandemic, summing up NIBS' work rather well. This institute, which is one of Britain's best-kept science secrets, is supported through funding from the Department of Health, yet its standards work is essentially Britain's gift to the world, ensuring the safety of biological products and making a major contribution to public health worldwide. But it's not quite as altruistic as it seems, because one consequence is that Britain's scientists are included when important global health decisions are made. Standards, it seems, win brownie points, and international prestige and influence too. NIBS allows Britain to punch far above its weight on the global health stage.